Alright guys, welcome back to Underground Science, and in this video we'll be talking about the common pathway, or also known as the converging pathway, which is the last pathway of the coagulation cascade. Alright, so make sure to go and watch my extrinsic and intrinsic pathway where we talked about all of this good stuff. Alright, even more above that you'll see in those videos where we talk about factor 12, 11, 9, and 8, and 10, right, which we can see here. So in the extrinsic pathway video, we left off saying that thromboplastin activates the inactive form of factor 7 into the active form of factor 7, then factor 7 helps activate factor 10. Factor 10 and 5 form a complex known as prothrombinase, which you can't see here, but let me go and write that down. So this complex that factor 10 and 5 forms is called prothrombinase. All right, and we also said it's called prothrombinase because it's an enzyme that will catalyze a reaction of inactive form of factor 2 to the active form of factor 2. Factor 2, the inactive form of factor 2, is called prothrombin, and prothrombinase will turn prothrombin into thrombin, which is the active form of factor 2. And we'll see that in this video as the final or converging, or the common, it's called many names like final pathway, converging pathway, and common pathway, all right? We'll just go and refer to it as the common pathway. All right, so let's go and come here and let's go and write down what we have so far, leading to the extrinsic pathway and the intrinsic pathway. So we have factor 10 that's active thanks to our factor 9, right? Thanks to our factor 9 and our factor 8, many things, right? Platelet factor 3, calcium, even factor 7. And so let's go and write that down. So we have factor 10 is the active form of factor 10 here, and we have a complex, and we have factor 10, which is also bounded, or it also has factor 5 that's binded to factor 10 as a complex. So we have factor 5 here, and let me go and write that 5 a little better. All right, so we have factor 5, which is forming a complex with factor 10. And together, the whole complex is known as prothrombinase, all right? Prothrombinase will then eventually go to thrombin, right? Or in particular, prothrombin, and convert that to thrombin, I should say. So this whole prothrombinase complex will then come all the way over here and find prothrombin that's floating in the blood. All right, we'll come here and find prothrombin. And remember, prothrombin is in the zymogen form. So it's in the form of a zymogen. And all a zymogen means in this case is it's in its inactive form. All right, so let's just go and write that down also. Prothrombin is the inactive form of factor 2. All right, so it's the inactive form of factor 2. Prothrombinase, or factor 10 and factor 5, also we can call it, will go ahead and activate the crap out of prothrombin, and it will turn prothrombin into the active form of factor 2. So remember, this is the inactive form because it's a zymogen. It will turn prothrombinase will turn prothrombin into thrombin. And thrombin is now not a zymogen because it's not inactive. It's going to be the active form, right, of factor 2. So the active form of factor 2. Oops, sorry about that random dot. The active form of factor 2. All right, now what happens next in the common pathway? All right, so let's see what happens next. So now that we have thrombin, there's many things that thrombin will do, right? Thrombin is like awesome, all right? So thrombin first will go ahead and activate factor 1, all right? So factor 1 is in its inactive form called fibrinogen. Fibrinogen is also a zymogen because it's inactive. So let's go and write that out. So thrombin will come here, find fibrinogen, right, fibrinogen, and it's going to activate the crap out of fibrinogen, turning it into fibrin. All right, and thrombin is going to do this. Now, fibrin was the whole goal of these pathways, because we have to make fibrin in order to have fibrin strands that hold the platelet plug together. So the platelet plug isn't loose and just goes to a random place and clots and gives us a heart attack or stroke. So fibrin will be responsible for holding / securing the platelet plug. All right, the platelet plug at the site of injury of the blood vessel. All right, but we need something else because the fibrin is going to be responsible for, it has to cross-link, all right? It has to cross-link and form strands that cross-link around the platelet plug, all right? So we need another clotting factor to do that. Well, what's going to happen is that thrombin will also go ahead and activate factor 13, all right? We'll also go ahead and activate a clotting factor known as factor 13. All right, so let's go and write that down. So the thrombin will activate the inactive, or so turn the inactive form of factor 13 into the active form of factor 13. Then the active form of factor 13 will then come here and it's going to cross-link the fibrin into fibrin strands. All right, so then what's going to lead, so the result of factor 13 acting on the fibrin is that we're going to have cross-linked fibrin strands. All right, and then these cross-linked fibrin strands, which is what we needed, will then go to the platelet plug. So let's say the platelet plug is right here, right? I'll save time and not draw the endothelial cells, but let's say the platelet plug, right? There's many platelets here that are like blocking the site of injury, so blood doesn't leak out into the interstitial space, right? So we have all these platelets here, and let's say, so now the cross-linked fibrin strands can go ahead and, so I'm going to go and draw more platelets here, can go ahead and actually show their shape here, like all cross-linked, right? So they're all cross-linked with each other, and you can see that these tough strands here are holding the platelet plug together, so the platelet plug doesn't move, right? It's holding this and securing the platelet plug at the site of injury. So let's go and label this. This is the site of injury. Site of injury. All right. So here we can see that thrombin is acting like a protease enzyme, all right? Because it's catalyzing these proteins like fibrinogen into the active form of fibrinogen, which is fibrin. So remember that this is the inactive form of fibrin. It's fibrinogen. It's an inactive form, all right? And then now thrombin acts as a protease, right? An enzyme that's going to catalyze this protein, this inactive protein, into the active form of fibrinogen or factor 1. So I'm going to write that down, right? So fibrinogen is the inactive form of factor 1, and then fibrin is going to be the active form of factor 1. So this is going to be the active form of factor 1. All right, so we can see thrombin is doing that. All right, and the reason why we have these fibrin strands that can actually cross-link and be better at its job is because of factor 13, right? One other thing that thrombin does it kind of helps itself in a way, all right, because it goes up here, right? It goes here, and it also activates the previous clotting factors that were involved in the extrinsic pathway and the intrinsic pathway, all right? And it goes ahead and activates factor 5. It stimulates it stimulates the reaction of converting the inactive form of factor 5 to the active form of factor 5, and it also helps convert the inactive form of factor 8 into the active form of factor 8. All right, so it also does that so that more thrombin can be produced later on, all right, in the common pathway. And one thing else that I should mention happens is that once we start getting a lot of thrombin production, right, a lot of thrombin synthesis, there's an enzyme or a protein that's formed in our liver, right? So our liver forms this. So let me go and attempt to draw liver again. Again, it doesn't look so good, but our liver kind of forms this, and it's called antithrombin. So our liver synthesizes antithrombin. Antithrombin will then go ahead and inhibit the catalyzation reaction from the converting of inactive form of thrombin, so prothrombin to thrombin. So it's actually going to inhibit prothrombin, the reaction of prothrombin to thrombin. So then less thrombin will be formed because we don't want too much clotting, or else it can get big enough and just clog that blood vessel as a whole. All right, so we need some negative feedback loops. So the antithrombin, you can think about it, acts as a part of a negative feedback loop when we're talking about the coagulation cascade. All right, so it acts as a negative feedback loop. All right, so that's that, right? So we pretty much finished everything that we needed to talk about, and we'll definitely talk about in the next video how we'll see a TPA, right? Tissue plasminogen activator. We'll talk about plasminogen and plasmin, and then that will be about how this clot right here can then break apart, because we don't want this clot just to stay here, right? Dangerous things could happen. Once once endothelial cells have healed, and more importantly, once blood has stopped trying to flow out because of our fibrin, right, our insoluble fibrin, we'll see how this platelet plug breaks apart with the help of a protease enzyme called a tissue plasminogen activator, and with the help of plasminogen and plasmin, mostly plasmin, all right? So that's pretty much it. That's all I want to talk about in this video. And one thing else that I just wanted to mention is that, it's mostly because of a hereditary or genetic disorders where you get hemophilia, right? So hemophilia is pretty much where you don't clot blood easily. So you have very much trouble in clotting blood. So trouble clotting blood, which is really dangerous because you can just bleed out really quick if you get even a small injury that, pokes all the way into your blood vessels or your skin, right in your dermis layer. So trouble clotting blood, and it's mostly hereditary. And so what happens here is that in the three types that we see hemophilia, right, in specific hemophilia A, B, and C, hemophilia A is associated with a factor VIII deficiency, all right? So hemophilia, crap, those random dots keep on appearing, I don't know what to do. So hemophilia A is associated with a deficiency in factor VIII, all right? So now that we know our coagulation cascade, we're familiar with factor VIII now, right? Remember, it's bound to factor IX that helps activate factor X. Hemophilia B, so hemophilia B is results in or is caused by a deficiency in factor IX. All right, so deficiency, I'm just going to write in a random different color, deficiency in factor IX, one of our clotting factors. And lastly, we see that hemophilia C, right, so the third type, hemophilia C is associated with a deficiency not in factor X, right, so crap, I forgot the C. Oh man, all right, so hemophilia C is not associated with the deficiency in factor X, but it's associated with the deficiency in factor XI. All right, so that's it. That's all I wanted to get through in this video about the coagulation cascade. We got a really good idea of it. And in the next video, we'll probably look at how the clot breaks apart, all right? Because we need that clot to break apart once it's done performing its job. All right, so we'll talk about it in the next video. I hope you enjoyed this video. I hope this video made sense to you guys. Hope you guys like and subscribe and hit that notification bell. I'll be making more videos in the future about cool topics in biochem, physics, and chem. And so see you guys later, and stay safe.